All right, so the title of my talk is the developmental tools for multiphoton imaging in mouse spinal cord neurons during locomotion. So, outline of my talk, I'm going to give you a bit of
Did he just completely drop off? What?
Yeah, I think he completely fell off.
I still see.
I'm sorry, did it disconnect again?
Yeah, it disconnected again. I'm not sure what happened.
Make sure I'm in share screen.
We know imaging spinal cord is really hard, so yeah.
Yes, it is.
Even the presentation.
It's a lot of
the
moving.
presentation is a lot of moving parts. All right, can you see my screen again?
Yeah, yep.
All right, cool. Okay, let's try to get through this. All right, so here are the basic setup for the talk. I'm going to go through the three main tools, which is a 3-photon excited microscopy, adaptive optics, and automatic kinematics detection, and gait detection. So yeah, this is an old slide, but basically the importance of locomotion in animals and humans is basically the bottom line is survival. So at the core of locomotion are central pattern generating circuits, and these are different types of neurons that have rhythmic oscillating output. So a lot of this older work has been done in invertebrate model systems, which I used to work on using a sharp electrode electrophysiology, and other people in leech, Cancer borealis crab. And in these small neural circuits, we're able to basically understand how these small central pattern generating circuits work. The thing is, in the mammalian system, a lot of these central pattern generating circuits are in the midbrain and the spinal cord. And we're going to be focusing on the spinal cord. And in normally for locomotor activity, descending electrical and modulatory excitatory input is given to these spinal cord circuits in the CPG circuits in the spinal cord to generate different types of behavior. And so I mentioned back in, well, people still do this, but back in the old days, in order to understand how spinal cord CPG circuits work, you need to use patch electrode recording to get the electrical outputs of the different types of CPG neurons. So you'll use ventral group recording using suction electrode and patch electrodes to basically see what the central pattern generating circuits, how they correlate to the ventral root output. So this is essentially similar to the connections to the limbs. But that's all well and good. But the thing is that this is done in in vitro preparation, meaning that the cord is outside of the animal. So it's kind of useless to us to understand if we're trying to understand how CPG circuits work if the circuit is outside of the animal. So what we need to do is to so this is a bit more background here. So there are different types of these central pattern generating circuit neurons. For example, V1 and V2B interneurons help to secure left-right alternation. I'm not going to go through all this because I know we're going to get into the optics, but this is important to understand because there are different types of the neurons that are located deep in the spinal cord. And we're going to need tools to see deep into the spinal cord. So the first thing we need to do is to develop a way for optical access in the spinal cord. This is the initial version of the window developed by Farrar and al. In 2012. And what this is basically a stainless steel window that's able to be implanted over the spinal cord. And we're able to, right here, this is the dorsal vein. And all this white part is where you see white matter and gray matter in the spinal cord. And so now that we have optical access, this is an anesthetized setup. We're able to look at the mouse spinal cord structure as well as the vasculature when the mouse is anesthetized. But the title of the talk is awake imaging during locomotion. So I'm going to get to the awake imaging soon. But this shows what 2-photon versus 3-photon looks like in the spinal cord. So I initially mentioned, well, here it's basically evident that, well, not basically evident, it's pretty much evident that 2-photon is not as good for, I shouldn't say, I don't want to insult anyone, but the 3-photon is actually a lot better to see deeper structures into the spinal cord. So here's the scale from 0, which is the surface, to about 500 microns deep into the spinal cord. And here we can see not only deeper in the spinal cord using 3-photon at 1330 nanometers, but also the resolution is a lot better. So I'm going to jump back a little bit. I mentioned the interneurons, for example, the V2As, V1s, etc. They're actually a lot deeper into the spinal cord. The surface of the spinal cord has the sensory neurons, and the CPG neurons, which are the V1, V2A, V2Bs, etc. They're actually responsible for patterning the different types of locomotion. So I'm going to get to that a little bit later on, but how do we get, these lovely images of the spinal cord here? So what we need to do is to develop a microscope. So this is a home-built microscope. I didn't build it. It was Mike Lamont. He's one of the optics experts in the lab. And what we need to do is not only just build a regular microscope, we need to implement adaptive optics. So the main premise of adaptive optics is to compensate for aberrations, optical aberrations in the spinal cord. Because what happens in the spinal cord is that there's a lot of myelin on the outside of the spinal cord compared to the brain. The brain has the gray matter on the outside, which is a lot easier to see through optically because there's a lot less optical, basically a lot less optical aberration. Because in yesterday's talk, we talked about how myelin is quite scattering because of all the lipids from the Schwann cells that are wrapping the spinal cord and wrapping the axons. And that will generate a lot of third harmonic generation. But in any case, so just a few more aspects of this microscope setup. We have a tilting objective. So the tilting objective is very useful because of the curvature of the mouse spinal cord. We're able to look at the lumbar region of the spinal cord, which contains the CPG circuits for moving of the hind legs. And with the tilting objective, we're able to have a much better view of those circuits while the mouse is spine fixed on the treadmill. So at the core of adaptive optics is basically a deformable mirror. I'm sure most people know what that is, but here's basically what a deformable mirror does. It has pistons on the back of it that's able to modify the shape of the mirror, and that's what we use to compensate for optical aberrations. And the imaging setup here, a quick question, can you all see this screen from
Yes,
Zoom?
with the cursor. Yep. We can see that.
I keep having to move it around. Let's minimize that. Okay.
I have the exact same issue. It was very hard to tell on my screen what people could see. But yeah, we see it moving.
Yeah. Okay, cool. All right. So we have a basic 4-channel detection optics setup where we're able to see TSG, green or yellow, and red or second harmonic generation. So under normal imaging day, we'll have to reset the deformable mirror and tune the different Zernike polynomials in order to tune the shape of the mirror. So what the Zernike polynomials do, as I said, they change the shape of the mirror. And because of the curvature of the spinal cord, there's when we tune the shape of the mirror, we're able to enhance the signal, which we are not able to see when from signals that are a lot deeper. And yeah, I mean, that's basically what the deformable mirror does. And then in the end, we'll simply maximize the signal amplitude that we get from imaging.
Is it closed loop or you doing it manually?
Yeah, go ahead. I'm sorry?
Is it closed loop or are you doing it manually and just looking at the image as you adjust it?
No, we do it. Yeah, we do it manually.
We do too.
There are other groups. Yeah, there are other groups that are working on a closed loop system to basically automate the way automate the optimization of the signal. When we're imaging at a particular depth, all we do is go at that particular depth, tune the Zernike polynomials and get an optimal signal before we start imaging.
Okay.
Yeah.
A related question. I'm sorry to interrupt, but what do you usually find the dominant aberration terms are? Is it kind of astigmatism and spherical, or it depends very much on exactly where you're imaging the spinal cords or where the curvature is and so on?
So it depends on where and how the mouse is oriented under our scope. But I've seen the Z5 vertical astigmatism and vertical coma. But as you get to the higher order Zernike polynomials, it does help, but it's mainly the astigmatism. So yeah, a lot of the tuning of the deformable mirror has been done in conjunction with another graduate student, Cheng, who recently graduated. He taught me most of the optics stuff. So we kind of been working together as a team on this very challenging project. And so I'm the one who's, along with some other students, is going to be going forward with this. Another aspect of this microscope is this final iris here. And so what happens with 3-photon imaging, there's a lot of heat that's generated at the surface from imaging at a long time. So this is a regular galvo setup. So at the turnaround of the mirrors, at the turnaround of the beam, sorry, this slow part here is going to cause a lot of burning on the edge. So we have an iris in order to mitigate that. Because what will happen if we're trying to image and we see the increase of the GCAM signal, it could be looked at as activity, but then that could actively burning. So there are a few times where we're sort of fooled into thinking that, wow, we see an activity at this depth when it's actually burning. All right, so now that we have the imaging setup, which is the deformable mirror 4-channel microscope, let's go back to the Laminectomy and Chronic imaging window. And just remind you again what it looks like. The window goes on top of the spinal cord, and we have these bars on the side that keep the spinal cord stable in place to mitigate motion. And this is just an example to show that even after a couple of days post-surgery, the mouse is able to walk, rear itself, and basically go on as normal. All right. So what we'll need to do, so one of the main aims of this project is to understand how the central pattern generating circuits in the spinal cord coordinate locomotion. So what we'll need to do is to film the movement of the hind legs and the activity, and then in the end, what we get is a few different data streams. One would be the calcium signals from the microscope, and then one would be the video from these 3 different cameras. And I'll get to later on, another set of data is the coordinates from the motion capture setup. And so here's one of the older prototypes of the motion capture setup. And we have three main cameras, one for the left and right side of the mouse. We initially had a circular treadmill, but we upgraded to a linear treadmill. So here's some of the initial set of data taken by Yiting Cheng. Here we have some of the CAMK2 GCAM neurons that are flashing during the mouse's locomotion on the circular treadmill. On the right here, we have calcium signals from these different ROIs as the mouse is moving on the treadmill. So we're able to see, using 3-photon in awake imaging, that some neurons are able to function in the mouse as it walks. We would like to actually image a lot deeper because, as I said initially before, the surface of the spinal cord contains most of the sensory neurons that would encode signals from when, for example, temperature and when the mouse touches the foot on the treadmill, but they're not really the main neurons that pattern the locomotion. So when I say pattern, I mean when the mouse is able to go into left-right alternation gait, if it's able to tap its leg multiple times on one side, even when it's scratching, multiple CPG circuits are engaged during are engaged. So on the left here, we have the new upgraded linear treadmill setup. This is a mouse that's getting ready to be imaged. On this, where my arrow is here, we have the IR lights. We're able to illuminate the mouse as it runs under the treadmill. So all right, so let me just go back a little bit. There's a lot of work that went into generating the mouse line that has the GCAM6S labeled into the CPG neurons for CHOX10. So we currently use the off system AI148. We're able to cross our pre-driver lines to the AI148 or 162 to generate the GCAM6 labels into the CPG neurons. So we also have other neurons that we're looking at as well. So this is just an example of the label GCAM6S and CHOX10 neurons. And here's just an example of the mouse, a different mouse walking on a motorized treadmill. So we're looking at 2 different aspects. One would be voluntary movement on the treadmill, and another is movement on a motorized treadmill. All right. So I mentioned the automated limb detection. So what we need to do is first train the mice to run on the treadmill. This is an example of the treadmill over here. And then after the mice are trained, we're able to use the cameras to film the left and right side of the mouse, and then use deep lab cut, which is able to mark different points of interest on the mouse. And we're able to then output the CSV data to create a wireframe diagram over a different amount of frames, and then correlate that to what we see on the calcium traces. So here on the left, here's an example of the hip, knee, and ankle. Someone else's speaker. So the hip, knee, and ankle angles as the mouse runs in the treadmill. These are the dots that are tracked during a typical session on the treadmill. So this was another REU student, Kelsey Walker, who actually one of the first students who helped me with this. And as I mentioned, the wireframe diagram, we were able to then output that and put that into our final pipeline to answer the question of CPG neurons and their function during locomotion. So the joint angles this is just some raw data showing the joint angles that we get from a typical running bout. From the hip, knee, and ankle, we see that over time, the hip, knee, and ankle angles obviously change during locomotion. Yeah, this is summarized exactly. This is probably. All right. So on the typical.
You need to focus on your schoolwork during school time.
So on a typical.
Your leg is healed.
Typical mouse gait.
I know it is.
The mouse
I've had those injuries.
Actually has a swing and stance phase. So on the initial swing phase, we see a different change of angles versus the stance phase on the mouse has its foot on the plantar surface of the treadmill. Oh yeah, and so a number of different questions came up previously as to whether there's a significant difference between spine fixed and tail held mice. Because the current optics setup is that we only are able to run the mice on the treadmill as they're fixed on the spinal cord. Right now, miniscope can be used, but I don't think 3P has been worked out for that yet. We're able to see on the surface level, but in order to see the neurons much deeper, I don't think miniscope is optimized to work right now. But here, this is one of the sample data showing that there's no significant difference between spine fixing and tail fixing the mice as they run on the treadmill. So yeah, I should have put this before. So there's a lot of training that actually goes on. So when we implant the spinal cord window, we have to handle and train the mice in order for them to run and ambulate well on the treadmill. Because back in my initial set of slides, I showed you that the CPG locomotor circuits are important for survival and optimal behavior. So if we were trying to understand the CPG circuits in the spinal cord that are responsible for normal behavior, we need the mice to behave normally on the treadmill. So in some of the initial trials, when the mice were spine fixed on the treadmill, they would freak out, they would not ambulate correctly, they would kind of spread their legs out wide. So we would see some calcium signals, but it's not during a typical normal locomotor behavior. So a lot of the work has to go into making sure that the window's on correctly and the mice are trained correctly, so that when we do the awake imaging, we see whatever signals we see are the actual correct signals. All right, so So here is another part of the data analysis pipeline, which is the Gait Cycle Analysis. So after we get all of the calcium data, the camera data, we have to use an automated system in order to automate a system in order to label the gait. So Matt Isaacson is the one who's developing this part of the analysis pipeline. So what he's able to do is to extract the toe coordinates, hip coordinates, and knee coordinates, and then do an initial pass to correlate that with fluorescence change at a particular ROI. And in addition to that, we're able to bin the different gaits based on where the toe angle is, where the toe coordinate is, sorry. So with the Gait Cycle Analysis, one other thing that's I mean, I should say, one other thing that's pretty obvious is that what sort of motion artifact do you see when the mouse is locomoting on the treadmill? Well, there is some motion artifact in the rostrocaudal direction, because there is, even though we have the mouse spinal cord fixed on the side using the metal bars, the spinal cord itself can actually move within the vertebral column. So there's a few things that we can do to mitigate that. Is when we do the laminectomy, we add some extra glue, surgical glue on the outside in order to stabilize that particular spot. So how do we know that that works? Well, here we have on the y-axis here the displacement for the x motion, y motion, and a z motion over a particular time series. And on the right here, this shows the histogram of the displacement that we see over particular frames. So on the x-axis, the x and y, that's actually, there is some, but it's actually okay because we have motion correction algorithms that can correct for the axial motion. The z, we can't really do much about, but in terms of algorithms to correct that, but we've gotten to the point where, as you can see on the bottom here in this histogram, where there's a lot more of the histogram a lot more of the points under 0 microns. This just tells us that in the z direction, there's not much motion artifact as the mouse runs. So when I say the z, there's the surface of the spinal cord, and this is the head of the mouse, this is the tail of the mouse. There is some motion in this direction, but not much in this direction. For example, when the mouse bobs its body up and down. So that's actually pretty good. So that's one of the major things we had to overcome. All right, so this is mainly shows the same thing where Matt's code is able to gate. I'm sorry, not gate. Bin walking versus non-walking. So we're able to pretty much quickly put in the data stream for the calcium signals, the speed of the treadmill, and in addition to that, the coordinate data from the deep lab cut.
Hey, Kawasi, I have a quick
All right,
question.
this is, yeah.
So, so you're essentially using motion correction to get rid of all of the motion artifacts from walking. Have you considered using the deep lab cut gait data to try to regress out the motion artifacts from walking?
I don't know. We haven't considered that. No.
Okay. Well, I guess if it works, it works.
Yeah. So So this is a summary slide that kind of brings together everything. Yeah, it brings together everything from this whole project, where the goal is to understand how these spinal cord CPG neurons function during locomotion. So here we have different ROIs from a mouse that was from the chalk stem neurons as the mouse is running on the spinal cord, running on the treadmill, sorry. So each of these traces shows the fluorescent signal of different ROIs correlated to the gait cycle. In this particular mouse, we did not see any clear evident activity of any clear correlation between any change in fluorescence and the gait cycle. This is just one example. In Yuting's defense, in his defense presentation, he actually has one good trace where there was the chalk stem neurons were correlated. Some of the chalk stem neurons, not all, some of the chalk stem neurons were correlated to actual footfall. So in that data, that's going to be coming up in the manuscript that we are putting together. So yeah, just to summarize, we have the detection optics setup, which is deformable mirror, new microscope setup. So yeah, deformable mirror is used to compensate for the optical aberrations in the spinal cord, including the curvature as well as the myelin. We have a deep lab cut, which is able to help us easily trace different points of interest on the mouse's hind limb. And in addition to that, we have the treadmill, which is able to give us a lot more data on the speed and acceleration of the mouse as it runs. So I'd like to thank people in the Schaefer Nishimura lab, Matt Isaacson, who's the postdoc who's helping with putting together the pipeline for the different data streams. Mike Lamont is the one who built the new microscope and built and did all the coding for the deformable mirror and the GUI. Yuting Cheng is the graduate student who I worked really close. We worked really close together to basically get this whole system going, where we have the mouse with the labeled spinal cord neurons, getting the window implanted, doing the imaging, the training, and then I'm the one who's mostly doing all the deep lab cut getting all the deep lab cut training data, and yeah, etc. There's quite a bit of time me and Yuting spent on this. And Daniel Rivera, who's the one who's been helping a lot with getting all the systems set up. Not all the systems, the synchronization between the scan image as well as our camera computer, because we have different computers running a lot of this stuff. Let's see, Samantha Krause is one of the RE students who started the deep lab cut. Okay, well, she didn't make deep lab cut. That's okay, I forgot their names. Yeah, it's going to come back to me later. And Kelsey Walker, who's one of the students who I worked with in order to start the awake locomotion imaging on the circular treadmill.